great pleasure to welcome you today at the Breakfast and Science meeting of the CIBM. Jonathan Wirsich, research scientist and operational manager of the MRI UNIGE Cognitive and Affective Neuroscience section of the CIBM. Jonathan is an expert of multimodal imaging, and today he will give us an overview about his research emerging EEG-fMRI and diffusion MRI imaging.
So, Jonathan, please, the stage is yours. Thanks.
Okay. So, hi, I'm Jonathan. I'm the operational manager in the CIBM section in Geneva. And we will talk a little bit about connectivity, and then I will talk a little bit about the other activity that we have in the lab. So if you think about brain connectivity, you can think of this also a little bit. Cars driving around in a city. So the city would be the brain, and streets connecting the different locations in the city will be the connectivity. So if we think of brain connectivity like this, we can think of something like, for example, on the weekend, everybody wants to go to the beach, and then they will all cross by the same crossing, and then there will be some kind of network hub where we have probably more connectivity, and then there will be like some exterior parts where we have less connectivity. So when we think about this in the brain, our measurements to do this non-invasively in the human brain are quite limited. Like we have diffusion MRI where we can do tracked overview feed, but this basically only gives us the number of lanes we have. We don't know how many cars are driving across these lanes. Probably if there are lanes, there should be a lot of cars also. Then we have the bold signal, which is really indirect. We're not really measuring the car. We are more like measuring that the car needs to stop from time to time to get some fuel. And then finally we have the EEG signal. So this is like basically putting on top of the brain some electrodes. It's like measuring like on top of a city, the smog or something the cars do. So yeah, so already to say quite limited if we want to go to the scale of neurons. So yeah. And this is my research interest. I'm interested how those different connectivities interact between each other. And the main thing that I'm looking at is functional connectivity. So we would look at the dynamic brain rhythms of two different brain regions. And then we calculate some kind of statistical dependency between those signals when we do resting state normally over 20 minutes, and then from this we can derive some connectivity measure. This connectivity measure, of course, exists because we have in the white matter some underlying fiber bundles that connect two brain regions, so they can synchronize from a point of view of functionality. And when we do this a little bit more systematic, we can create a so-called connectivity matrix where now each dot in this matrix signifies the connectivity strength between two brain regions. And then if we add a third region here, then we get another dot in this matrix, which now is connecting 1 to 3, and then we can fill up this whole matrix with our connectivity measure.
This is overhead.
All right. Okay. So when we think about different modalities, so now we can think of, okay, how can we derive different modalities, and can we push them through the same brain atlas to get the same connectivity matrix for the different modalities? So as I explained to you earlier, the main modalities that we have is EEG, functional MRI, and diffusion MRI. For the EEG, we have really good temporal resolution from 1 millisecond, but then, as I told you, we put the electrodes on the scalp. So when we want to reconstruct this activity, we don't get a really good spatial resolution, so around 1 centimeter or higher. For the functional MRI, it's the other way around. We have a better spatial resolution of 3 millimeters normally, but then we need 1 to 2 seconds to get a picture of the whole brain. So yeah. So this is already a trade-off. Maybe we can combine this to get the best of both worlds. And then finally, for the diffusion MRI, we also have the resolution of 3 millimeters, but then we have no dynamics at all. We only have the underlying structure. And then we can think of a framework where we push everything to the same atlas, and then we can look how the connectivities interact between each other to better understand the brain. So one of the first studies I did back in Marseille was to acquire in the context of a clinical study some participants that were sleep deprived. So they came in, one day got a resting state, then they stayed awake all night, and then they got another resting state the next day. And it was to test the effects of Alzheimer medication on sleep-deprived participants. And yeah, so we had the EEG-fMRI recording before and after the sleep deprivation. So of course, once we put them into the scanner, they fell asleep directly on the second session. So we had quite a lot of sleep data available. And what we showed here is when you do the sleep staging. So we use the EEG that we record simultaneously in the MRI only to get the spindles and the sleep events to stage the sleep. And we use this as a covariable in our connectivity analysis of the fMRI. And what we showed is when we do this, we can extract independent networks both of sleep and also of the medication intake. So this is one example how we can use EEG inside the MRI scanner. But it's really basic. Like you only use it for sleep staging because you cannot do this in fMRI directly. So yeah, if you want to go a little further, so I didn't talk at all about all the different correction methods that we have when we do acquisitions simultaneously in the EEG-fMRI. So basically, this is only the clean signal that you're getting, but of course there's a lot of interactions between the gradients and the fMRI when you do this simultaneously. So you should only do this when you really want to have simultaneously activity. Otherwise, it's better to do the EEG outside the scanner and then combine the fMRI in a separate experiment. I won't talk in detail on the correction methods today. So let's just assume that the EEG is clean. So what we can do now is we can reconstruct the system of the electrodes of the EEG into the space of the brain, and then we can directly connect this to the fMRI. So let's just assume we have some algorithms that can derive from those electrode signals activity in each brain region. And then we do the same for the fMRI. For the fMRI, of course, it's very easy. We just take the time course and we do the average across the regions. And then for fMRI, we normally use Pearson's correlation or something similar to do the connectivity matrix. And then for the EEG, we have the problem that now when you do the source reconstruction, there are a lot of correlated signal because like there's the problem of so-called source leakage, that you don't know exactly if some signal, because you have a really smooth signal that you're reconstructing, some signal is bleeding into other regions. So you would get really high connectivity. They're only correlated because like you're basically reconstructing the same signal. And how you would go around this is to take only the signal that is different in phase. So you will just assume I'm only interested But in the statistical dependencies that are a little later, some milliseconds later than the signal that I'm getting. And we can do this by using, for example, imaginary part of the currency, and then we can also derive some kind of connectivity matrix for the different rhythms of the EEG. So here we can directly profit from the high temporal resolution, so we can split up the EEG signal into different frequency bands. And when we do this, we have shown, but also other people have shown both in fMRI, EEG, and MEC that those are all correlated around 0.4 when we do the correlation between those matrices. This is true for all the different frequency bands. So like the correlations, they change a little bit, but they stay all around 0.3 to 0.4. If you have any questions, feel free to stop me, because like I think we have enough time. Okay. So with this in mind, we now try to push this a little bit further. So we were asking like, okay, we have those EEG connectomes and we have those fMRI connectomes, and they're somehow correlated, but is there also some information in there? So they're correlated at 0.3. So of course there's some information that in there that's not the same, but is this information relevant in a sense of neuronal brain activity, or is it just noise and we cannot recover this? And to address this problem, we now tried to predict the underlying structural connectivity by combining in a linear model both the EEG and the fMRI. And the idea was the fit should be better if there's a complementary information, because then the linear model will put together the both informations that then can predict the underlying diffusion. And then we're kind of sure that we have some signal that's not artifactual, but it's only due to the EEG or to the fMRI. Always in terms of, okay, to predict the diffusion MRI. So we still make a little error, but what we showed there is that, yes, when we use EEG and fMRI, we can really do a good fit of the diffusion MRI. So you can see the fitting here. But in the end, what we also showed in this paper is that most of the connectivity is explained by the distance anyways. So yeah, I mean, you might not scan at all, and like you explain already half of the connectivity. So yeah, so there's additional information, but if you don't want to spend too much money, you also can just take the distance between different brain regions, and then you have a rough estimate. So yeah, from that we were like, okay, yeah, we're not really interested in this rough estimate. Can we also show like that the dynamics are linked? So this really, here we really need now simultaneous recordings, because like until now, of course I showed simultaneous recordings, but basically you could have done the EEG outside the scanner and the fMRI, in the scanner, of course. But here now, if you want to look at dynamics and to look if the dynamics are correlated from window to window, then you really need to do this simultaneously. You cannot do this, otherwise you need to find kind of states. So this is more difficult. So that's where EEG-fMRI can really shine. And that's what we basically shown in some work we did back in the University of Illinois. So we took some EEG and fMRI data and we split it into different time windows. And we showed that when we take the mutual information between those different sliding windows, that we get some significant mutual information as compared to some randomly scrambled connectivity matrix. So yeah, that means that those dynamic functional connectivities are somehow linked. This also validates kind of the discussion of if there's dynamic functional connectivity or not in fMRI, because like now we have like the proof from the underlying EEG that there some part of the dynamics are linked. Of course, it's not as easy as it seems. The mutual information is quite little. So in other papers, we also shown that when we use intercranial EEG-fMRI and EEG-fMRI in multi-layer graph models, that most part of the EEG and fMRI dynamics are quite different, even if they are stable or reliable inside each modality. So yeah. So the next step I was interested in when I came to Geneva here is to now, what I talked about was always little cohorts of 30 participants or something. And so now we really tried to get together some bigger data set, still quite little. So we took 74 healthy subjects undergoing EEG-fMRI, both at 1.5 Tesla, at 3 Tesla, 7 Tesla, also using different cap layouts. So for example, 64 electrodes EEG, but also 256 electrode EEG. And what we were able to show back then is that the correlation between EEG and fMRI connectivity remained stable. So what I show here is the beta connectivity of the EEG versus the functional connectivity of the fMRI. So you can see here at the 1.5, we get the best correlation. So that probably means that the functional connectivity of the fMRI is already good at 1.5 Tesla, but we have much lesser artifacts at the 1.5 Tesla on the EEG. So the replicability of the EEG was much better when we do this in a low field scanner than as compared to the 7 Tesla, where we get really some nonlinear artifacts to correct. So yeah, we also use this data set now to look a little bit in the reliability of classic EEG-fMRI experiments that look at EEG power versus functional connectivity. And again, the results where it's not that easy, probably. So it can be like, there's a lot of difference in the literature, and that's probably because if you use different data sets, you get quite different results. So yeah, this just came out in Imaging Neuroscience. It's in press, so it should come out soon if you're interested. So the next step in Geneva that we did was that we were working on epileptic patients. So now we tried to use those properties that we have in the correlation between EEG and fMRI. And we asked ourselves, does this change when we use, for example, epileptic patients? So, I mean, we can extract the difference between functional MRI and EEG in epilepsy for the patient separately. But is it also the relationship between EEG and fMRI that changes? And we did a little study before back in Marseille looking at the relationship between diffusion MRI and functional MRI. And what we've shown is indeed the relationship between diffusion MRI and fMRI is much more correlated in temporal lobe patients. And this effect seems to be also the case when we look at theta and fMRI connectivity. And what you see here in the controls, we have the standard 0.35 correlation that we get when we correlate the EEG and fMRI, but in right temporal lobe patients, this is much higher. This was only the case in right temporal lobe epilepsy, but not in left temporal lobe epilepsy. So there's like some discussion ongoing if those epilepsies, they seem to recruit much different networks, even if they're the same type of temporal lobe epilepsy. So yeah, so the next step I showed you earlier that some of the data sets were done at 7 Tesla. So the next idea was okay, when we do 7 Tesla, then we can also go to much higher resolutions. So here you see the cortical gray matter. So we have normally 53 millimeters of thickness. So I told you earlier, we have a resolution of fMRI of around 3 millimeters. But yeah, if you want to tap into different layers, you cannot do this at 3 tests. So yeah, but once you go to 7 Tesla, you can go to 0.8 millimeters or more. And then, yeah, you start to be able to try to separate those different layers in the brain. So this is work spearheaded by Schwal, which we did together in Bern. So basically, Schwal, he developed new prototype for the special EEG cap that can go into the special antenna of the 7 Tesla. So I don't know if you know a little bit the setup of the 7 Tesla. So the antenna is quite, there's not a lot of space. And also if you want to connect some EEG, there's not some space to get the cables out somewhere. So you have like, I don't know, 1 centimeter maximum to put the cables through. So Schwal was developing something really special so you can put all the cables through at 7 Tesla, so we can scan with the newest equipment. So this is some data from the 7 Tesla data Schwal acquired before, and that we also used in the paper that I showed before. So what you can see here is that on the old 7 Tesla scanner here in Lausanne, the signal to noise quality was quite limited. So the idea was to push this a little bit further. So those are the recording that he did in Bern. So yeah, so really we can go from this a little bad SNR picture to some laminar fMRI while doing EEG simultaneously. And the status of this project is, for now, that it's confirmed to be safe. So we validated that there's not too much heating. We don't have too much SNR penalties. And the EEG quality is good enough to do some connectons. This work is in review currently in imaging neuroscience, but you can also already check out the link in bioarchive. And you probably know the paper already, it's 15 hours working here. So now, when we look at this, when we have some 3 millimeter data with concurrent EEG-fMRI, we get something like this, and you see here the 0.8 millimeter sequence that we now have. Much better, but you see also down there's still some work to do on the frontal lobe. So yeah, that's what we're currently validating is how much of this signal down here can you recover to do some concomitants, or do you just need to throw away down the frontal lobe and develop new sequences to adapt for this? So that's what we're currently looking at. This is one of the prototypes that Schwal developed. So you can see here the really special cable that needs to fit in there, and like it's quite tight. So good luck to people who want to do this with patients. So yeah, we also work together with Brain Project. So they already developed a commercial prototype that has been acquired for the CIBM and also for the campus biotech. And we're currently piloting a study to adapt this approach to in the context of a sleep study, just to get the difficulty a little bit higher. So the patient needs to be stuck in there and some extra. So let's see what comes out there. So yeah, so the final goal, of course, for my project is to now get connections on different layers. So now, if you do a really rough cut in the surface and the deep layer, you can easily do this. Of course, this is stupid because like the main activity of EEG and fMRI, you would expect it in layer 4, so it's directly in the middle. So maybe you need 3 layers or another better protocol. So like, but just assume this is a reasonable model for now for demonstration. So now we have a data set from Bern. At the same time, I trained a postdoc in Marseille to do some extra acquisitions, and now the data needs to be analyzed. But this is the basic idea. To now add to the framework that I proposed to you, using diffusion MRI and EEG, now above deep and the surface layer connectome that we can now then correlate between those different measurements that we have. To go one step farther and a little bit back to my research that we did in Marseille. So here you can see really noisy EEG data before cleaning. So that's what you get when you acquire data in EEG-fMRI. But let's just assume you can clean it. And what we showed in this study when showing both famous and unfamiliar faces is that when we clean the data and then we take all data time courses of the EEG and we do an ICA, we can extract different ICA components on the whole group and project those components back into the subject space. So what we get here now is those different ERP components that we have when we send a visual stimuli. So there's a quite big negativity on 170 milliseconds, then there's another negativity on 400 milliseconds, and positivity at 600 milliseconds. And with those ICA components, when we project them back onto the subject space, now we get a really clean single trial estimate of the peak of each trial. And this peak we can then use to correlate this with our fMRI. So basically, at each trial, so this each time point here is one trial, we correlate this to the voxel activity we have in the fMRI. And when we do this, we basically get the voxels that are activated at this time point in each. So it's a kind of correlation. It's not a direct causality, but it basically gives you the voxels that are correlated to this time point in each. And what we've shown in 2014 is that when we do this, we kind of can get occipital to frontal activity when we go along in time. So this method kind of seems to work to get some activity. Of course, it's also quite noisy, as you see here. So yeah, the final idea is now, can we do this on a layer basis? Like basically now we would be able to tap into sub-second events of the EEG and correlate them to multi-layer activity in the 7 Tesla. And this is then we could use to better understand, for example, face recognition, feedback loops that are supposed to be on different layers, but also quite close to each other in time. And also for epileptic events, for example, which are quite fast and we can measure them in fMRI, we can take this framework to extract the dynamics of those kinds of fast millisecond activity to tap into the multi-layer activity of the fMRI. So that's any questions so far?
Maybe we keep the questions at the end.
If you can remember. So I will please go into some miscellaneous stuff that we also do at the EPFL, CISA, and CIBM. So currently with Serge at the we are getting out a free data set created by eBrains. It's in print in scientific data, so it should come out soon, but already available in eBrains. So basically we get some EEG data with epileptic spikes. So those are the epileptic spikes I talked about. So you see here, now probably you don't see, but this is basically 1 second of activity. So the dynamics are quite fast of these epileptic activities. And here we propose a data set where we have basically a prostration, a T1, and also the surgery region where the patient was operated. And now you can look. So this is the first data set where you could validate if I take this spike and I project this back into the brain, do I hit the surgery region? So optimally, if the patients have good outcome, I should have hit this region. So this is the kind of data set where you can validate your source reconstruction algorithms with, or also to better understand why in patients where the surgery didn't work out, so maybe we can work on that a little bit. Then there's another project I've been involved in, the connector mapper from Shield, so headed by Patrick Hartmann. where you can automatically do your connectors for diffusion MRI, and with Daniel, we started to use this also back in Geneva. Let's see if it's something coming out. So yeah, if you want to do your connectors automatically, I recommend to use connection mapper. Then for the development, we have in Geneva a little bit. So we try to adapt some GABA blast protocols, so maybe I will get in touch with the gene soon to validate this. But basically, the human sequence seems to work. Then we also explored if there's any use of multi-echo fMRI. So the idea of multi-echo fMRI is that you get a TR a little bit longer, but you acquire with different echoes, and you probably get a better SNR, and you also get better signal to noise and frontal regions. So we validated this in an olfactory experiment. You eliminate those frontal regions of the olfactory bulb. And what we found out is if we do multi-band acquisitions or when we do multi-echo sequence, we get kind of the same activations. So for now, I would say no need to do some fancy modeling on the multi-echo, but we will continue investigating. But for now, you're fine with your multi-band fMRI. And then there's also a bigger problem. We're a project where we try to assess a little bit the common data quality to develop common data assurance protocols, just to make sure that everybody uses a good sequence for their specific fMRI study. Then, beyond fMRI, we're also working on in collaboration with Emily and Sébastien, and also from our team, we worked on photodiodes that can measure directly on the screen in the MRI room the onset of the visual stimulus that we have. So we get rid of all the lag that we have communicating via parallel port triggers. And we're currently applying this to an EEG-fMRI study where we really want to do ERP studies inside the scanner. So normally in fMRI, you don't care if the stimulus is like 1 hour milliseconds later, because like the MRI will take 1 second to acquire anyways. But yeah, once you want to do ERPs, we need to get more or less exact timing. So that's one project we have ongoing. And if you have an idea how to use this equipment, feel free to reach out to Emily. Then we're also working on applying the new EEG caps that they have from Brain Products, which have a carbon wire loop. So the idea of the carbon wire loop is that it's a closed electrical circuit that picks up all the noise in the scanner, so the movement induced inside a magnetic field, but also like the vibration artifacts we get from the pump. And we can use this signal and request this out of our EEG. And later we want to use this in a neurofeedback experiment. So we're currently working on correcting the EEG online inside the scanner with using this carbon wires. So the idea basically is that this activity will give me all the noise that I can request directly out of my signal, and what you can see here so far is that when we do the EEG outside the scanner, eyes open versus eyes closed, we have a nice alpha peak, and then when we do the carbon wire loop correction online, we can also get this alpha peak. So now, this is really rough. Data quality check, I guess, won't be that clean for the other things, but yeah, in principle, this works. So yeah, this we then want to adapt an EEG-fMRI experiment where we do a neurofeedback both of the fMRI signal and also on the EEG signal. If you're coming to the fMRI research day of CIBM next week, already, then you can also feel free to check out. There's an oral presentation from Ricardo Gali on the fMRI-based neurofeedback of the study. For the EEG part, you still need to wait. Maybe for the next EEG day. So yeah. Then just to tell you what equipment we have back in Geneva, so in case you have some funny ideas, we're always happy to talk about how to use this. especially the driving system that just stands around for now. So we have an fMRI compatible driving system, some gamepad. We also just installed last year 3D screen with polarized lenses. So basically you can do 3D experiments, but at the same time you can keep your eye tracker and everything that you have in fMRI, because like it's basically on the screen and behind. So what you can see here, then we also have some MRI compatible only for 3 Tesla cameras to do, for example, emotion recognition or gesture recognition inside the scanner. So yeah, if you have any ideas how to use this, feel free to reach out. And otherwise, again, on the CIBM Research Day, Chi-Song will present the first results of an experiment using the 3D screen inside the scanner. So with that, sorry for the CISA video people, I couldn't put everybody, but I guess you know who we are, so I put only the international collaborators. So thanks everybody who contributed so far, and so thanks everybody who will contribute in the future. And I'm happy to have your questions.
Thanks a lot.